Thank you, Luis. I'm really delighted to be here, and I want to thank the organizers for the opportunity to present this work. I'm especially glad to talk after Philippe Nesser and Nicolas Vauchelet. Philippe gave wonderful explanations on what gene drive is, and Nicolas explained an analytical method I'm going to use. So hopefully, thanks to their talks, I will be able to finish in time, and I am acutely aware of the importance that I finish in time. My talk will be quite classical, an introduction, 2 parts, and a conclusion. In the second part, I will present work that has already been published, when you can somehow get rid of heterozygotes in your gene drive system. And in the third part, I will talk about recent progress on what we will have to do when there are heterozygotes. What is gene drive? So as Philippe explained yesterday, gene drive is basically any kind of genetic control that allows you to force the fixation of an allele in a population. So contrarily to Mendelian classical population genetics, with gene drive, you ensure that the allele is fixed in the population. I'm not a biologist, but in the last decade, gene drive has been made possible thanks to CRISPR-Cas9. And basically, the idea is that the drive allele is going to suppress the wild-type allele on the other chromosome, replace it, and then you have converted the heterozygote into an homozygote individual. There are two times, in the life cycle of heterozygotes when this conversion can occur, depending on what promoters you use. On the left of this strange figure, you have the case where the conversion occurs early in the life of the heterozygote, as soon as the zygote is formed. On the right, you have the other case, basically, where the heterozygote lives on, has an heterozygote individual, and then only produces drive gametes. So it's a late conversion in the germline. And what you need to observe right now is that there are 2 special cases where you can reduce heterozygous populations. The first case is when you have conversion in the zygote with 100% chances of conversion. In this case, you have actually no heterozygote in the population. And the second case is the case of perfect germline conversion with dominance of the drive allele. In this case, all heterozygous individuals will produce 100% drive gametes. And because the drive allele is dominant, they will live their life as heterozygotes. And so you cannot distinguish at any level, at the phenotypical level, you cannot distinguish heterozygotes from drive homozygotes. And so in the first part of the talk, I will focus on these 2 special cases where you can either say that there are no heterozygotes or say that heterozygotes are just like homozygotes. But first, what would we want to do such a kind of genetic control? There are mainly 2 kinds of applications. The first one is evolutionary rescue or biodiversity conservation. And in such a case, the drive is called a rescue drive, and its goal is to increase the population size. And on the other hand, you have pest control strategies, where you use the gene drive either to replace the population with a new population, like in the Wolbachia strategy, or let's say more radically, you use the drive to suppress a part of the population, or maybe even to completely eradicate it. Because of the many risks associated to population suppression or eliminations, the experiments have been quite limited, as Philippe explained yesterday. They are all done in cages, and even if a cage can be quite large, it's never going to reach the size of the classical diffusion rate of a mosquito, let's say. So that's why mathematical modeling is of the utmost importance regarding gene drive right now. And that's also why countermeasures are actively developed, but I'm not going to talk about countermeasures today. There is a way to distinguish drives, another way, that depends on the release threshold. So in the first case, you don't have a release threshold. And let's say one drive mosquito escaping the lab is going to start an invasion, trigger an invasion, and then it's going to be very difficult to stop this invasion. In the second case, you have a release threshold. You need to release enough mosquitoes, or many individuals, and then the invasion will start, and it's actually going to be easier to stop this invasion. Why would there be a release threshold? Because you have a competition between the biased inheritance due to the drive mechanism and to the fitness cost. And mathematically, this release threshold problem is going to appear as a monostability versus bistability question. In any kind of model you can think of, like discrete generations, continuous generations, discrete space, continuous space, you're going to find monostability versus bistability. And so the question that we ask is basically, how do these two typologies interact? So you have the effect-based typology, rescue, replacement, suppression, eradication, and you have the threshold typology. And for instance, is it possible at all to make threshold-dependent eradication drives? Because of course we want to make threshold-dependent drives in order to be able to stop invasions. And so that's the first work we did. So suppression or eradication drives with logistic growth in space with reducible heterozygotes. I'm going to use a reaction diffusion model. And by that I mean that the variation of the population is going to be equal to the net migration plus reproduction minus mortality. The population density is a function n of t and x. X is typically 1D or 2D or 3D. The population variation is the time derivative of the population density. The net migration is going to be an isotropic diffusion, and I will comment on that in the end if I have time in view of Philippe's talk yesterday. The mortality is going to be a linear term, and the reproduction is going to be a more complicated term that I'm going to try to explain right now, but I'm not a probabilist. So bear with me. In the population, you have 50% of males, 50% of females at every point, at every time, and for every genotype. So no sex structure. And then you say, okay, I want to know how many individuals carrying the genotype capital G I'm going to have in the next generation. And this is the probability of, so you need to sum over all genotypes for mothers and for fathers, the probability that these two genotypes are going to mate. And then you want to know the probability that the descendant, the offsprings, are going to carry the correct genotype. And then you want to know the size of the number of offsprings, and then the probability that these offsprings are going to reach adult stage. So the mating probabilities, you can decompose them as the probability that the parents meet and then that they reproduce, and the probability that they meet, we assume that it's proportional to the frequency in the population, and the probability that they mate is just the product of their fertility rate. And then where the gene drive appears in the calculation of the probability of the genotype, knowing the genotypes of the parents. Usually you would do the classical tables that we all did long ago for Mendelian inheritance. But here we have gene drive, so that's going to change the tables, basically. And so you obtain the reaction diffusion model with two alleles, so O for the wild type and D for the drive. You assume that convergence and dominance rate are such that you can get rid of the heterozygous population. So you have only two population densities to track, NO, the wild type population density, and ND, the drive population density. And you assume that basically the wild type population density, when it is all alone, undergoes the most classical logistic growth possible. And then you measure the phenotypical effects of the drive through a diffusion rate, a fecundity rate, a juvenile survival rate, and a death rate. And you obtain these two horrible equations that are actually not so horrible. So in the first line, you have the diffusion term for the wild-type population, the mortality rate, the mortality at rate 1, the number of offsprings in each generation with the competition due to logistic growth, and then the probability of making wild-type offsprings, which is proportional to the frequency of wild-type individuals in the population. And then you have the same thing on the second line, so the diffusion of drive individuals, their mortality, their reproduction rate, and the probabilities that individuals meet and mate. I'm going to simplify this system by assuming that selection only acts on juvenile survival, so all other rates are equal to 1, and the juvenile survival is written as 1 minus S, and S is the fitness cost, as in many talks this week. And so you have the simplified system, which is written here, and the first thing you can do is look at what happens when one population density is fixed. So, for instance, if you have only wild-type alleles, you recover the classical Fisher-KPP logistic diffusive equation. And on the contrary, if you have only drive individuals, then you have an equation that looks the same, but that have basically two issues, so two outcomes. If this quantity R minus S minus SR is non-positive, then you're going to have eradication of the population, suppression of the population. And on the contrary, if it's positive, then you have a logistic growth with carrying capacity equal to 1 minus S over R1 minus S. And I want to comment on that right now. You see that the carrying capacity is going to be 0, or the drive population is going to go extinct if S is close to 1, or if R is close to 0. So that means that if the fitness cost is too large, the drive is not viable, and if or is going to go extinct, and if the growth rate is too small, same thing. You can rewrite the system in proportion density variables, because that's what we do in population genetics, and if you do so, you obtain this system that you might recognize from earlier talks this week. I want to draw your attention on this advection term with the gradient of logarithm of n. So actually, by going through to the proportion density variables, you simplify the reaction terms, and you don't have the possible 0 denominator anymore, but you have this gradient of logarithm of n, which is also not defined when n is close to 0. So in both systems, you have a problem when you are close to eradication. What can you say about this system mathematically? You can verify that it is well posed, and actually it's not, but it is if the wild-type population initial density is uniformly positive. You can study the stationary states, spatially homogeneous, and the two most important stationary states are the one with only gene drive and the one with no gene drive. And you might want to do stability analysis on these stationary states, but the problem is that you cannot do it for the eradication state, which is the most interesting one. And also you cannot apply any kind of monotone dynamical system theory, because you have two regimes in these two systems, actually, a competitive regime and a predative regime, and so you cannot apply any kind of classical theory for Locta-Volterra systems, let's say. So the systems are complicated, and it's natural to try to simplify them. And the case where you can simplify them is the replacement case. So when do we have replacement in such a system? We have replacement when the drive carrying capacity is close to 1. And this occurs either if the fitness cost is close to 0. So that's the weak selection assumption. But actually, in our case, it means that the drive and the wild type are phenotypically identical, and so it's not really that interesting. Or if you have very fast Malthusian growth, so R is close to infinity. And actually, if R is close to infinity, you can repeat what Nicolas presented this morning, and you obtain this decoupled bistable equation on the proportion of drive in the population. And there is no gradient of logarithm of n anymore, so it's fine. The ratio is not very convenient, so you can try to say, okay, it's close to the cubic equation that we saw earlier this week. But again, the fitness cost is typically not so small. So this approximation is just qualitative because it's again a bistable equation, but the thresholds are going to be different. The natural questions are the validity of these two approximations, in particular if time is infinite, because the method of Nicola only works in a finite time horizon, and for suppression or eradication drives, because again, this applies for replacement drives. If you simulate numerically this system, what do you see? You see in 1D that, for instance, if you introduce some drive on the right, it's going to grow. So this is population densities, not proportion. The drive is going to grow and then to invade at a constant speed with a constant profile. But you also can find cases where the drive invasion is going to fail. And actually, the drive is going to completely disappear from the population, and the wild-type population is going to invade again, actually at its classical Fisher-KPP speed. So there are cases of success, depending on what you call success for gene drive, and cases of failure. And so it's natural, in view of these simulations, to consider traveling waves with constant speed. And to study this traveling wave is mostly to study the sign of the speed, and to study the sign of the speed is to study the competition between these three forces. The gene drive tends to push towards the left. The natural selection is in favor of the wild type, so pushes towards the right, yes. And the gradient of population generates an ecological invasion that also pushes towards the right. And so what we want to do is to try to say things about on the sign of the wave speed c as a function of the Malthusian growth rate r and the fitness cost s. You can plot the same thing in proportion density variables. And depending on your school, it might help you or not. And then you can try to analyze these traveling waves. And but there are important issues. The first one is that the traveling wave with no gene drive always exists. It's the Fisher-KPP traveling wave for the wild-type population, and it's always there, and there is a half-line of such traveling waves, so they are there. The second problem is that the non-trivial traveling waves, so the ones with drive, are observed numerically in many cases, but not in all cases, as I showed earlier. And so there should be some parameter range for existence, for stability, and for selection against trivial traveling waves. And finally, there is again this singularity close to the eradication state and the mixed monotonicities, which mean that the usual tools to analyze traveling waves for reaction diffusion systems fail. So actually, you cannot do much analytically on these traveling waves. If I want to compare these traveling waves with the simpler system where you have a population replacement, and I focus on the cubic equation, which is simpler. So for the cubic equation, the state where you have only drive, so p equal 1 is stable. The state where you have no drive, p equal to 0 is stable also if and only if the fitness cost is larger than 50%. So if the fitness cost is larger than 50%, you have a threshold-dependent reversible drive. The sign of the speed is completely known, actually, for the cubic equation, and it's exactly the sign of s minus 2/3. So if s is smaller than 2/3, you have invasion of the drive, and if it's larger, you have reinvasion of the wild type. And so the good region, so the region where the drive is variable and reversible, is between 1/2 and 2/3 for the fitness cost. Obviously, on this heat map, nothing depends on R because there is no effect of the population dynamics. There is only population genetics for this replacement wave. And I want to say that if you take the full equation with the ratio instead of the cubic, it's just going to change the S equal 2/3 into S equal 0.697 something. So it's very close. If you consider the full system, on the contrary, you have this more complicated heat map where the good region, the good central region is reduced. And the region that is increased, on the contrary, is the non-viable region. There is also this new eradication solid line below which you have eradication of the population after the invasion, after the drive invasion. And interestingly, there are two regions in the non-viable eradication case, but I'm not going to comment too much on that. What I want to say is that analytically, we are able to characterize the sign of the speed on the left of this blue curve and on the right of the brown curve. So typically, we are not able to find the correct, the precise location of the zero level line, but we know that, well, I mean, we still know the sign in a large range of parameters. And how do we prove this, how do we construct this blue curve and brown curve? The first element of proof is to notice that you can actually quite easily produce a result on non-existence of non-trivial traveling waves. So if you have a non-trivial eradication wave, you know that the drive population density is going to go from 0 to 0 through a global maximum. And so you can look at what happens at this global maximum, and after very basic computations, you find the contradiction if R is too small. So if R is too small, depending on S, you have no non-trivial traveling wave. And so this means that the only traveling wave is the Fisher KPP wild-type traveling wave, and you know the sign of the invasion, the direction of the invasion. When you assume that you have indeed a non-trivial traveling wave, and again, the existence is a completely open problem, but if you have one, then you can use the monotonicity between the monotone relation between the total population density n and the proportion p. You can use some change of variable from a paper by Grégoire Nadin, Nicolas, and Martin Schregarek to have an explicit characterization of the sign of the speed. Explicit, not so much, because it depends on this function h, which is the relation between n and p that we don't know. But if you are able to estimate h, you are able to estimate the sign of the speed, and this is what we did. So as a summary for this second part, on the left, you have the case of replacement waves where you neglect demography, population dynamics. So you have on the left of the left figure the irreversible drive that you want to avoid. On the right of the left figure, you have the non-viable drive that have no interest. And in the center of the left figure, you have the reversible viable drives. But they are all replacement drives. On the right figure, you account for demography. You have this new red zone for eradication, and basically the same the same free zones, but the nice zone, let's say, is smaller. And in particular, you can see on this figure that there is indeed a zone for threshold-dependent eradication drive, but it's a very small kind of triangle at the bottom of the figure. And if you try to think of like spatial heterogeneities or stochastic fluctuations or whatever, you understand that it's going to be very difficult to actually construct a viable, reversible eradication drive. Now I'm going to talk about more recent work in progress with Vincent, Florence, and a PhD student, Lena, Klay, on the case where you have irreducible heterozygous. So in the case where you cannot reduce the system to a two population density system. I want to remind first this figure. So on the left, so you have this life cycle of heterozygotes. On the left, you have the case of conversion in the zygote, early conversion. On the right, you have the case of late conversion in the germline. And again, the two cases that I call reducible are the cases where you have full conversion, and if it's germline conversion, if you have dominance of the drive allele. So when you are not in one of these two reducible cases, what do you obtain? You have a new system with two new parameters. So parameter C, which is not the speed, which is the conversion rate, as in Philip's talk yesterday, and a parameter H, which is the dominance rate for the fitness of heterozygotes. And you have also a new population density for the heterozygous individuals. I'm not going to change the other assumptions, so specifically, selection only acts on juvenile survival and populations grow logistically. Once you make these assumptions, you obtain for the zygote conversion this horrible system. So as you can see, it's so horrible that it does not fit in the slide. Anyway, you can verify that if C is equal to 1 and if the heterozygote population is initially at 0, it's going to stay at 0, and you recover the previous system. On the case of germline conversion, you have again a nightmarish system, and again you can verify that if the conversion rate and the dominance rate are both equal to 1, you recover the previous system, this time by assimilating heterozygous individuals to drive homozygous individuals. So these systems are horrible, but we know special cases of them, at least. You can repeat this, the numerical analysis and obtain these nice heat maps. I want to draw your attention on two things. First, in the previous part, the zero level line for the speed was a kind of convex curve. Here it is a straight line, and that's really mysterious. The other thing I want to draw your attention on is the fact that level lines seem to be either vertical or horizontal. And that's completely counterintuitive because it basically means that depending on the region you choose, you can forget about one of the two parameters, R and S. But actually, that's a completely wrong way of thinking of the problem because you need to remind yourself that below the eradication curve, you have eradication waves, and at the top, you have replacement waves. So even if the speeds do not depend on R and S, the profiles strongly depend on R and S. So these waves are definitely not dependent on only one parameter. What can we do analytically? We can reduce the two three-component systems to two-component systems by considering the allelic densities instead of the population densities. And once you have these two new two-component systems, you can repeat the transformation to have a proportion, frequency, and total population formulations, so PN formulations. Once you have these systems of two equations, you can repeat the maximum principal argument for the non-existence of eradication wave. And then the thing we did with these two systems that we did not do before, is to look at two asymptotic regimes. So the first one is R goes to infinity, so that's the replacement regime, and that's where you recover equations of the type of Martin and Nicolas. And the other regime is R close to 0. And actually, it turns out that if you look carefully at the system when R is 0, at least formally, you find a kind of SIR system with a frequency-dependent transmission. And we know these systems actually, I'm not going to say quite well, but much more than these horrible gene drive systems. So at least formally, we can study the limit R equal to 0 and R equal to plus infinity. If we do so, having in mind that we would like to have this kind of vertical level lines, we find the quality of the speeds at R equals 0 and at R equal infinity under some horrible conditions. But these conditions are like explicit and we can check them. Comments are that first, in the two special reducible cases, these horrible conditions reduce to S less than 1/4. And this condition is known. It's the condition for the cubic equation, for the Schroeder-Vosley equation to be KPP equation. So the speed is linearly determined, and it's 2 square root of the f prime of 0, and it's known. And it's equal, actually, to the linearly determined speed at R equals 0. These conditions are sufficient, but not necessary, because it's a KPP type criterion, and they are monostable waves that are pooled but not KPP. So, but we heard enough about pooled and pushed font this week. As I said before, the speeds might be equal, but the profiles are definitely not. And this is a first hints towards the verticality of level lines in the SR plane. As a conclusion, and I'm going to be right in time, that's wonderful. If I can summarize, so we have reaction diffusion models for gene drives impacting population size, whether it's suppression or total eradication. And actually, you could use exactly the same systems for rescue drives if you wanted. We see the importance of strong selection, that is a S not close to 0, because this is where you can have eradication, for instance. Regarding the rigorous analysis of traveling waves, we are able to find non-existence results, speed estimates, and formal limits. And we have these nice numerical simulations in 1 dimension. What about what Philippe said yesterday? So Philippe said that diffusion is not ideal to model these eradication waves because you have chasing effects. And actually, we did a stochastic simulation with Florence in our paper, and we also observed this chasing effect. But there are 3 reasons for which diffusion is still relevant, in my opinion. The first one is that the heat maps we obtain for stochastic simulations when the carrying capacities are sufficiently large is actually exactly the same as the one we obtain for the deterministic model. The second reason is that the mean time to eliminate a new colony formed by this kind of incursions in the wiped out territory is going to depend in some simple way, I guess, on the deterministic wave speed. And the third reason is that by eliminating chasing effects, so by considering the deterministic system, you're actually looking for the best possible scenario for the drive. And so it gives you kind of a per estimate on lower estimate on what you might want to estimate. And so, in my opinion, it is a very interesting perspective to try to bridge the gap between these deterministic traveling waves that I presented and the stochastic chasing effect that Philippe presented yesterday. But I have no clue on how to do it. And to conclude, a few more perspectives. So, of course, regarding traveling waves, we would like to have existence results. We would like to have things to say about their multiplicity, about their stability, about their monotonicity. And it would be nice to make the formal limits r equals 0 and r equals infinity rigorous. It's always interesting to find better speed estimates, but on this, I'm not really hopeful. And we can also look at propagation in higher dimensions with spatial heterogeneities. Can you block the invasion with some kind of wall or holes or whatever? And finally, it's a question raised by numerical simulations. Can we prove this verticality, this strange and mysterious verticality of level lines in the SR plane? That's it for me, and I'm right in time. Thank you for your attention.
Okay, thank you for this nice talk, Leo. Any questions in the room? Yeah, Nicolas.
Thank you for the presentation and for the very interesting results. I just don't understand very well how you compute the good region for the system. What you call the good regions in the...
Here.
A little bit before. Yes, this one.
How I compute it?
Yes.
Okay. You can verify that, okay, so it's related to the way we compute the blue and brown curves. On the left of S equal 1/2, you can verify that you have a pooled wave. And so it's a pooled wave and it's not going to be reversible, etc., etc., etc. So bad region. And then you can improve the blue curve thanks to the estimates with the h to the power 4 that comes from your paper. But you can hope at least that on the on the right of S equal 1/2, it's going to be in some sense bistable. It's not going to be bistable because you have the eradication case where you cannot compute the stability, but still. And similarly, on the right of the brown curve, you can verify that you have waves that are going in the wrong way, and so it's non-viable. And this time you know numerically that on the right of the dashed curve, waves are going in the wrong direction, and so it's non-viable. So, I mean, the S equal 1/2 is actually a conjecture, like kind of conjecture for the monostability versus bistability threshold. But it's, I mean, if you look at the linearization for the ND for the drive equation, you see that it's going to be monostable if S is larger than smaller than 1/2 and bistable otherwise. So you have the correct sign for the f prime of 0, but you cannot study the stability. It's kind of frustrating.
And the function h in your situation, you have an idea to find them, to find this function?
So we know it's a decreasing function from the interval 0, 1 to this interval. And actually, for the moment, we have no more estimates. But we are able to say stuff about the monotonicity of h to the power 4 times r1 minus h plus 1, which is amazing. And of course, what you would like to do is say that, for instance, h minus h of p is between, let's say, 1 minus n to some power and 1 minus n to some other power. But in order to do so, you would need to apply a comparison principle in this system that has no comparison principle. So it's a completely open question.
Okay, thank you. Yeah.
Excuse me, just curious. So the chasing effect, so is the dimension important, or is the stochastic important to see chasing effects, or is both like necessary?
So we only did 1D simulations.
So this already.
Yeah, and in 1D, what you see actually is that if I take this eradication wave, for instance, you see that some drive individuals, so some green individuals, are going to make stochastic incursions in the right territory, going to grow and make a new colony, and so the drive is going to go backwards and eat them again, and etc., etc., etc. I don't know about the importance of dimension, but I guess that you might find answers in Philippe Messer's paper. Probably dimension is important, probably, because it's related to Brownian motion. And,
Okay, any other questions or comments online or here, Vincent? No? Okay, so let's finish nearly in time, I think. Let's thank all the speakers of this morning.